I'm Bob Hergenrother. I'm the Senior Director of Research and Development at Biocoat. I'd like to talk to you about strategies for reducing friction in medical devices and the ways you go about evaluating those. So the benefits of lubricity in medical devices are several-fold. The first part is ease of access of these devices. If you think about it, the doctor has to be able to get to the site in order to be able to do any sort of therapy. And there are twists and turns in the bodies. So much so that in some of these devices, especially like in a neurovascular device, these imparting the lubricity on the medical device improves the functionality of the device, so that you could actually, it's almost impossible to get to the site without that. In other words, if you think about it, the vessels are kind of fragile, and so that they can be irritated. And by minimizing the friction, you can minimize the trauma to those vessels, so they don't spasm and closing off the blood flow to further parts of the body. And finally, you're shortening the procedure time. This has benefits both for the patient in terms of time on the operating table, anesthesia, even radiation exposure, but also for the medical workers as well, the doctors, nurses, technicians. Again, their operator fatigue, they minimize that, as well as minimizing radiation, and then just overall shortening the time in the OR. So the requirements for lubricous surface are several-fold. Obviously, you want to have low friction as you're moving these devices through the body. But beyond that, there's also you want good durability. If you think about it, the body has very few straight shots in the body. And so there's twists and turns and different access points where you have to kind of select which way you want to turn. So there's back and forth, and so you want that low lubricity to maintain that same value throughout the procedure time. Those were sort of the two main areas of evaluating lubricity up until probably about, I'd say, 10 years ago. And since then, there's been this other piece of these low particulates. So it's been sort of driven by some of the regulatory agencies of asking, any particles that are shedding, tell us why those are not causing a problem and why they're not causing an impact. Talk about the strategies for reducing friction. You can, if you start from this, you can go from the beginning, choice of materials of making your device. If you think about it, a low durometer PBAX material is rather tacky and soft compared to, say, a PTFE material, which is very, very slippery, but it does not have some of the good bulk properties that you may want. And so, but you can choose within that some that have both inherent bulk properties and surface properties that would work out. A lot of times, though, you're wanting bulk properties that impart greater strength, stiffness, that would not translate to a lubricious surface on that surface. And so then one way to do this is with the extruder, adding some compounding additives into there. Which are sometimes waxes or other extrusion aids, which can bloom to the surface and then modify sort of lower that surface energy, and so you've got a more slippery surface. The other final way of doing it, which is probably more near and dear to my heart, is coatings on the surface, where you're actually separating out the materials that are using this structurally from the coating materials. And so, for instance, in a Biocoat material, we put a base coat on that bonds very tenaciously to that substrate, and then from there, covalently bond our lubricious hydrophilic top coat. For evaluation of lubricity, there's a few parts of this. There's kind of two ways of doing this. One is what I kind of call a pad test, where you take two sort of rubber pads, squeeze the part together, and pull that up between them under tension, usually under in the hydrated solution. And you're putting sort of applied force, and then you're measuring the force it takes there, and that should be considerably less. Sort of best in class in a lot of these cases are at least 10x reduction from that applied force, and even best in class is 20x. That gives a direct comparison of the different coatings and really minimizes sort of the device design. So it's a good way of comparing different coatings, but then if you want to look at in terms of thinking about it for that device, getting through to simulating what's going into the body, you'd have what I call a tortuous path test, which is a simulation of the anatomy in the body, for example, like a neurovascular anatomy. That looks at both the device and the coating together, and you can evaluate that. And in seeing how well that ease of insertion, you can measure the force that's taken to apply that. The other part of that is then, so that goes for the initial friction. Then if you're doing multiple cycles on that, you can look at the durability for that device. Particulate evaluation is the next piece of evaluating coatings. Originally, sort of that lubricity and durability were sort of the main criteria of evaluating a coating. Recently, regulatory agencies have been asking for test results and basically justification for particulate levels from coated devices. Here's the problem, is that there's multiple factors that can contribute to particulate generation. There are the coatings, but then there's also just the device design, how stiff that is. And then the ancillary devices, there's the sheaths, there's the guide wires, even the wipes that are on the table. All those generate, can do the particulates. And so you have to kind of separate that out to really look at from what we're talking about in terms of the device itself. The challenge is there are standards and guidances for coatings, but none are really entirely applicable for evaluating particulates from medical devices. Now, there are particulate guidances from agencies such as like the United States Pharmacopoeia. There's one of particulates for injectable solutions, USP 788. This defines limits in solutions, so a vial that is being injected. And they recommend looking at particles and totaling them up from particles that are greater than 10 microns as well as particles that are greater than 25 microns. It does a fantastic job of giving instructions on how to count the particles. However, it is, as the title says, it is for an injectable solution. And so it does not really give guidance on how to generate that sample from a medical device into that solution and what would be considered an acceptable threshold for that. FDA has put out several guidance documents, one for PTCA catheters, another one for guide wires, where they recommend counting not only just in 2 different size bins, but 3, a greater than 10 micron size, greater than 25, and even a greater than 50 micron size, using an appropriate simulated use model. And that simulated use model is up to the manufacturer's discretion of having a relevant one. And then the other part is they recommend providing an interpretation of the particulate data. The challenge is they don't really provide what exactly an acceptance criteria is for that. So there is some little uncertainty for this, and we need to kind of, I want to kind of go through some of the challenges and considerations for doing the particle testing. The keys to particulate measurement are several-fold. As the FDA mentions, you need to use a simulated use model, and it needs to be clinically relevant. So, for example, ASTM has a model that is useful for stent retention. So for a cardiology device, you may want to use that model to track the device through collecting the flush solution afterwards. However, for example, in a peripheral model, you may want something a little bit more of a serpentine winding tortuous path. You really need to kind of define what that model is and why it is clinically relevant for your device and your coating. The other piece of this is you need to define the entire test procedure. Is that there are a lot of ways that you can change the values. And so, for example, defining the storage conditions that you're having your device and the solutions. The sample container, is that plastic, is it glass, how are you washing it? Flush solutions, how well you're filtering those, what that is that water, is that PBS? As well as even things like the duration of the time between testing for the particles and the time that you're actually measuring the particles. And then the detection method. USP talks about either light obscuration or impedance methods, and either one can work, but kind of defining what that is. The other key for this, as you can see from all of these different test conditions, you need to run comparator or control samples, because the value by itself you need to provide some sort of context for what that value means in interpretation. So for example, on the looking at two different models of generating particles. So we used this ASTM cardiology model and a serpentine tortuous path. There was a very good correlation when we took coated rods between the two. However, it's not a one-to-one. And so while the ASTM model typically has higher particulate numbers, it actually gives a nice correlation with this other model. And so either one can work for this, but you can't just interchange between the two. And so this is an example of how these, you get sort of similar results, but really defining the entire test system. So let's look at some additives and then finally some evaluations of hydrophilic coatings. And the first part is some lubricious additives that can be added to the extrusion to minimize the friction. And so this is an example of data that we had generated where we have some completely unmodified polyurethane and PBEX tubing, as well as ones that have sort of these low friction additives added into the extrusion. And then the other part is then putting a hydrophilic coating on top of them. So taking a look at all of these on these 2mm tubings, you can see that both of these additives do reduce the friction somewhat from their unmodified ones, about around 25%. However, as you do on cycling it back and forth 30 cycles, these have kind of bloomed to the surface. You can see that they've kind of worn off. And so by the end of it, they're about the same as an uncoated device. Then if you look at ones that we've applied as a hydrophilic coating on, one, you can see that there's a substantial reduction. So it's not just a 25%, it is a 95% plus reduction in that friction, and very durable throughout those 30 cycles. And what was interesting is that both applying these as the coating on the unmodified tubing as well as these ones that have these extrusion aids, it actually was able to coat onto there and bind very well to all three of those substrates. So we want to look at the particulates of these devices or these tubings as well as compared to a hydrophilic coating. And in this case, let's start with the hydrophilic coatings. They have around total greater than 10 micron particles of about around 100 on these. And the ones with these additives, it's about twice that, around 200, which is not bad. But also keep in mind that from the lubricity, these were incredibly difficult to insert into this tortuous path to generate these particles. So looking at, in terms of evaluation, you need to have both the lubricity and the durability and the particulates all in mind as you're looking at that and kind of optimizing for all three of those. So the other thing we did was look at the various hydrophilic coatings. And we did this by actually purchasing neurovascular microcatheters from the manufacturers. Two of them actually had Biocoat coatings on them. The other four had other hydrophilic coatings. And so again, we looked at these from both the lubricity, the durability, and the particulates. So let's look at the lubricity and durability first. And in this case, you can see that most of these catheters actually have similar lubricity results on these, around 15 grams to a little less than 10. All of these, again, evaluate on that pad test. We're applying 470 grams and pulling that on. So it's a dramatic reduction in the friction. There was one that had some, one of the catheters that the durability was not quite as good, as you can see that changed. But most of them were pretty good in terms of the durability as well as the lubricity. And if you think about it, this is how most people evaluated coatings up to 7 to 10 years ago. You had the lubricity and the durability. But when you look at the particulate piece, it kind of changes the interpretation of these coatings. Looking at the particulate evaluation for these hydrophilic coatings, we're kind of looking at it two different ways. One is looking at the total particle counts from these microcatheters. And so it's number of particles greater than 10 microns for the entire device. And you can see that there's a rather dramatic difference between the various coatings. The one that was that catheter C4 which didn't have much of a great durability, also not terribly surprising, has some rather high particulate generation. For some of the ones that were, say, with the Biocoat ones, had some very low particulates, and even some of the other ones had about twice that. But other ones also had some rather higher values, 10x higher than some of the best-in-class ones. So if you look at also on the particle counts, we compare it to coatings done at Biocoat on tubing compared to devices that were coated at the manufacturer. You can see that our coatings actually are very consistent from ones coated here versus at our customer's manufacturing thing. These are shown on particles per centimeter squared to kind of normalize everything. But again, the numbers are remarkably comparable, and then the other devices are 10 or even 20x higher than some of these best-in-class devices. Putting this all together, you can look at both the friction and the particulate generation on the same graph. And for this, it kind of really differentiates between the various coatings. There are some that are low particulates but then higher friction. There's other ones that have a lower friction value but higher particulates. And that has been traditionally sort of a trade-off between those two. Now there's ones that best-in-class ones, you can have both. Lower is better on both, that you can have both low friction and low particulates. In summary, what I've shown today is that friction reduction is a critical design requirement for many medical devices. And there's a variety of ways to enhance that lubricity. There's the choice of the materials themselves. You can do additives in the extrusion, or then do hydrophilic top coats on those devices. And also with this, that you need a comprehensive way of evaluating these friction-reducing materials, both in terms of lubricity, durability, and particulate generation. Now, we've covered a lot of material today, so I'd be happy to address any questions, and feel free to contact me.